guys, how are y'all?
I'm good. Have you heard people trying to reintroduce cheetahs to India? I'm not sure how they're going to do it with their low genetic reproductive success and low genetic diversity. There was a severe bottleneck that occurred 10,000 years ago, which makes them nearly all siblings. But the idea is intriguing.
I haven't, but that reminds me of this study I read recently about laparoscopic oviductal AI in domestic cats. Maybe they'll have some success with that method when it comes to endangered species. The study itself demonstrated high pregnancy percentages with low sperm numbers.
I think I also read that study. Is that the one that was published in like 2013? I thought it was a little invasive for like generalized all species AI, as it holds like the added risk of like the female going through like general anesthetic and the surgical like infection risk. But it seems like a potential option in species that are endangered or like really tricky to breed. Basically, like where the potential like benefits outweigh the risk.
So why did they investigate oviductal insemination?
Well, as the author expressed in the article, there's some concerns with the traditional procedure of sperm deposition into the uterine lumen. Reported pregnancy percentages have been low, and the method requires high numbers of spermatozoa for successful fertilization. And other common AI methods, such as vaginal insemination, have also shown the need for high sperm numbers.
Oh yeah, semen collection in felids can be super difficult. Professionals will often resort to general anesthesia and electroejaculation. Felids have a small ejaculate volume and a significant portion of which may be lost throughout the entire process from collection to insemination. Along with that, some species have less than 50% of their spermatozoa per ejaculate that are able to be classified as morphologically normal.
How does electroejaculation work? I don't think I've ever seen that in practice.
I think I've got a clip for that. If you give me a sec, I can share it. So it's just like a probe, and they use electricity to cause the ejaculation in the anestheticized male.
Cool. So what benefits do we get out of laparoscopic oviductal AI specifically?
Well, it improves pregnancy outcomes compared to the other methods by allowing the semen to be deposited in closer proximity to the site of fertilization within the oviduct. This allows the spermatozoa to avoid difficult transport through the uterotubule junction, reducing the number of motile sperm needed for successful insemination. That is extra important in cases involving species like endangered felids when sperm counts are low or there's a low volume of seminal fluid.
In addition to comparing intrauterine and intraoviductal AI methods, the study was actually also trying to see how using short-acting porcine luteinizing hormone, or PLH, as the ovulatory signal would compare to the typical usage of long-acting human chorionic gonadotropin, since HCG can cause some undesirable side effects. Here, I can send you the article. Take a look.
I will take a look and get back to you.
Thanks, me too. Let's call again when Mallory and I are done.
Hey, so I just finished reading. It was a pretty interesting paper. I see why they wanted to test an alternative to HCG. I didn't realize that HCG could lead to unwanted secondary follicular growth and ovulation. But that does make sense since HCG is a pretty large glycoprotein, so it stays in circulation longer.
Same. I didn't realize the specific difficulties of feline AI before. It was pretty interesting that they decided to test whether ECG PLH treated queens would be able to carry offspring to term following AI.
So how do they stimulate ovulation since laparoscopic AI bypasses the vagina altogether? Aren't felids induced ovulators?
Yeah, they're stimulated by movement in the vagina, causing nervous impulses that trigger the secretion of the gonadotropin releasing hormone. So GnRH then causes luteinizing hormone to surge, and the LH surge triggers ovulation. And stimulation occurs at mating, so if mating doesn't occur, then there's no ovulation. And the cycle repeats with progesterone remaining low. So how does AI get around that?
For that, there's one method is simply to insert like a cotton swab into the queen's vagina and mimic the natural stimulation that would occur during mating. This does the same thing like while triggering GnRH, LH, and therefore ovulation.
Yeah, you could also just administer GnRH or HCG instead, and that just triggers the same cascade. And if you use that instead of the manual stimulation on day like 2 or 3 of estrus, it also induces ovulation.
Cool. I think I have a chart that would help us visualize this timeline better. If I can just share my screen real quick.
Oh, so the LH surge is brought about, but that would have to be time flow. As we know, 3 matings in a 6-hour window has the highest chance of causing ovulation, and mating on days 3 to 4 of estrus will give us the greatest LH surge, with some individual variation, of course.
Yeah, then ovulation occurs 2 to 3 days after mating or 1 to 2 days after the LH surge. The oocyte then only survives for 2 to 4 days after ovulation, and this potential oocyte survivability period accounts for if ovulation occurred earlier or later in the 2 to 3 days after mating. So theoretically, the fertilization period is 2 to 5 days after mating.
But if the fertilization period is 2 to 5 days after mating, does that mean insemination happens then? As naturally, insemination occurs at the same time as a mating, right?
Right.
When that happens, the sperm survive long enough to fertilize the ovulated oocytes in that fertilization period, because by the time we're in that fertilization period, the queen won't stand to be mated anymore.
Oh, how come?
Well, after ovulation occurs, the progesterone levels increase, which causes the cervix to close. Also, the estrogen goes down, so they stop doing things like standing and showing signs of being in estrus.
I see.
Yeah, this isn't a problem for uterine or oviductal AI, but since we bypass the cervix altogether, especially since frozen and thawed semen don't last as long after insemination as fresh semen, we can just wait until the optimal fertilization period comes around and inseminate then.
So either way, you would do insemination 3 to 4 days after stimulation?
Mm-hmm. The insemination would be hard to do vaginally or transcervically, as the queen has increasingly narrow anatomy from vestibule to the cranial vagina. I think there's a good picture of this. I share this. Natural mating in felines creates enough pressure that deposited semen enters into the cranial vagina and easily passes through the internal and external cervical os into the uterus as an immediate passage.
Yeah, but standard AI practices can't really replicate that high pressure, so the majority of semen remains in the vagina. I mean, semen could be assisted by placing the queen in dorsal recumbency on a table and tilting it to 30 degrees, but the vagina is still really narrow, so we're also probably working with low semen counts.
Yeah, that's why in this study, they used surgical AI to inseminate both the uterus and the oviduct of each queen. The queens had an intramuscular injection of 100 units of ECG combined with either 75 units of HCG or 1,000 units of ECG 85 hours later, which were also given intramuscularly. AI was done 31 to 33 hours after the second gonadotropin injection.
The sperm motility and concentration were reassessed and the sample diluted in feline optimized culture medium with HEPs. A set amount of motile spermatozoa was used to inseminate each location, oviduct or uterus, within the female reproductive tract.
Speaking of, I have issues with the experimental group being made up of only 16 domestic shorthair queens and 2 domestic shorthair talls. Their age range of 10 months to 3 years was good, but to me, this is a very small sample group for a clinical study, and I'm not sure if it ends up actually being an entirely useful model population.
That's a good point, and I agree, but I did see a more recent clip from the Cincinnati Zoo that worked with this research project, and they showed further success with other felids. I can actually show you if you'd like.
In cats. So we can take vitrified sperm, which is frozen in very low numbers, and put it into the oviduct and still get fertilization and produce kittens. Now, with oviductal AI, we can use very low sperm numbers. It's not like doing a conventional AI procedure. Is that typically you need a lot of sperm to produce babies with AI. But with oviductal AI, we can use very low sperm numbers, sperm that's been vitrified, and produce offspring. So we have done some preliminary studies with semen from ocelots and fishing cats. These are 2 endangered cat species. It appears that vitrification works just as effectively with those species as it does with domestic cats. We haven't tried to produce any offspring yet with that vitrified sperm, but we have been able to fertilize oocytes in vitro using ocelot and fishing cat sperm that have been vitrified. That's what
domestic cats could prove to be a good model species in at least a few endangered felids. It's still a very limited sample size, but they did do quite a few statistical tests to analyze their data.
Yeah, the groups of cats were non-factorial design, randomized control group, trials, and meaning that, so non-factorial design is that there is no third group that has both treatments in this case, and since it's not applicable in this scenario, and they used a paired t-test as the same group colony or cats was tested under different treatments.
For analysis of the laparoscopic insemination sites, they used the McNamara test. This test is used to assess dichotomous variables, which are variables that have to fit in one of two groups, which are usually mutual. So in this case, a pregnancy cannot have been created by both sets of sperm at both insemination sites. It must have been one or the other.
The Shapiro-Wilk test and Anderson-Darling test were used together to ensure that the data was of clinical use and not skewed in a way that gives an inaccurate description of the population.
So the chi-squared test was used to compare what is observed against what was expected. So in this study, it compared the number of females that became pregnant by AI with ECG and PLH against the number of pregnant females treated with ECG and HCG, which was the standard AI practice.
And then they gave a confidence interval of like 95% and like a small group that does not make it in this paper. Like it just doesn't make the paper like very like clinically robust. However, like new studies into oviductal insemination support these findings, I guess. Yeah. By the way, did anyone else notice the two males that were used were related to each other? Male 1 was sire of male 2. This further limits like the genetic diversity in the study even further. I mean, I get that it was like trying to limit the confounding factors, but it makes the scope of the study incredibly narrow.
I saw it being a larger flaw potentially in differentiating the parentage of the fetuses in study 1 and the kittens in the second study.
But in addition to that flaw, I think genetic diversity would be important because if you're having two related males, then it could introduce that variable of, well, because they're related, both of their sperm have similar capabilities in terms of motility, survivability, all those things. And I think it would be better in a study to have unrelated males so that you don't have that bias towards stronger genetics.
But genetic diversity wasn't the end goal of this specific study because they're not working with an endangered species. They were demonstrating a model of an alternate AI method to potentially be used on an endangered species to be able to get successful fertilization with less sperm. And that allows them to select stronger sperm from the samples they do have in a smaller population of an endangered species. Which is another point. In an endangered species, you already have the limited gene pool, and even though you're trying to diversify, you only have so many options.
So it could be comparable to an endangered species study where you end up using related males.
Yeah, the point is figuring out if you're getting a more successful fertilization rate at a certain site. And then if you only have a certain amount that you can use from a singular male, you can be more confident and your end results will be more successful.
So it's like a two sides of the coin kind of thing. Like as a model for the real world scenario, it's realistic. But in a study where you're trying to get full solidified results and solid statistics, you would want the larger sample size.
Yeah, much larger. You know, I guess another factor that could affect the study is that all the queens were housed together in like a strictly controlled environment, which, despite scientists' best effort, could have induced stress in some of the queens, as they're not like very social, except in some like domestic cases, anyway. And as we know, stress can affect fertility.
Ultimately, the study concluded that oviductal insemination with ECG and PLH treated females might be preferable to uterine AI and the standard practice, but the sample size of study 1 in this paper was really small, like we said, and so restrictive that it doesn't really come to any like solid conclusions.
And that's just study 1 in this paper. I mean, study 2 was a mess. Only 3 females were used to try and produce live litters of kittens, and the sire of one litter was unclear, and the litter of another queen was unaccounted for.
I agree. Study 2 in this paper is currently clinically irrelevant as the results are flimsy at best. But study 1 and 2 need to be repeated with much larger sample sizes and with a wider sampling pool.
I agree. It should definitely be investigated further. The implications of a perfected method could be game-changing for endangered felids. A higher success rate to AI would decrease costs, workload, and resources required in repopulation attempts.
Definitely. It looks like March this year, there was a first successful AI of Tushima leopard cat using oviductal AI, and they used the PLH with the ECG. Also in 2017, the a ma, tiger, Pallas cat, ocelot, and the fishing cat were successfully inseminated via laparoscopic oviductal AI. And that was with low motor sperm.
It's getting kind of late.
you.
It was a good chat.
Well, anyways, have a nice one, guys. Bye.
Yeah.